Good afternoon. This is Dr. Dan Guerra from VerEvMed Studios in the Pacific Northwest. Today we're going to cover a topic which I've advertised on my Facebook page that's going to examine some of the neurochemical and neurocellular effects of cannabis on specific subtypes of the brain, particularly on arborization associated with both neurons and microglia in the central nervous system. Now, before we get started, I just want to put a caveat on here. The talk I'm giving today is not something that's generated by any kind of socio or political understanding of the fact that marijuana, which is, of course, the plant that has high levels of tetrahydrocannabinol, and that would be the bioactive substance we're going to be looking at today in these research papers. Nothing about the fact that it's being considered as legal for legalization in many states in this country. In fact, I think worldwide, we already probably know that. It's more about what we do know scientifically about the effects of this particular drug on the central nervous system. So I want to bring that out at the beginning. Now, I will mention some of the aspects at the very beginning about why people are studying this. But after that, it'll just be pure examination of the published literature. So let's get started. All right. Here we go. All right. So again, I'm Dr. Dan Guerra. There's my email. Contact me if you have any questions at all about this talk or any of the talks that I've generated for VerEvMed. Probably also you can see videos of me from when I was professor at various universities. Some of those are out there as well, I'm told. And certainly if you Google search me, you'll probably find those. The point here is that this is a VerEvMed production. That's our company, VerEvMed. There's our website. And there is our official email address that you can contact us to become a client. I'm not going to talk very much about VerEvMed today. That's all probably, except maybe briefly at the end. Only that I am co-founder and chief scientific officer, and that these talks that I'm giving as videos for YouTube are meant only as a representation or demonstration of the kind of thing we can do for you individually when you have a research or scientific question. You as a pharmaceutical scientist, or as a physician, or as the head of a hospital, as a nutritionist, exercise physiologist, or indeed as any normal person, layperson. All right, what's the topic today? Does regular cannabis use correlate with neuronal dysfunction? And the subtitle, THC treated murine prefrontal cortical neurons fail both structural and functional adaptation. Okay, so that's a partial answer to the question, I guess you could say. And we're going to see whether or not the data supports it. So again, there's my email address. Contact me directly. There's my Facebook page where all of these VerEvMed lectures are put up. I am in the process of putting them on my website, but I don't have any help doing this, so it's a time-consuming process that I hope to be able to get done sometime soon. This picture, actually, I should mention, it is right outside of my house, about, 1,000 yards away. And that's the Clearwater River flooding its banks in the springtime. This is a huge ponderosa pine, judging from the other trees that are there that I've cut down because they died because of this constant inundated and flooding that kills the roots. Get it? Arborization, which is what we're going to talk about today. It's probably between 80 and 120 years old, that tree right there. So that's pretty interesting all in itself. Now, what I want to say is that notice how this tree could be looked at as a neural system that functions in a healthy manner, in a physiological manner, and also in a temporal level. That is, it ages. And that organism, that ponderosa pine tree, will age and eventually it will die. Now, it could die prematurely based on the environment. Here you see the inundation of the Clearwater River, and that constant inundation, what does it do? It suffocates the plant because it submerses the root structure. If there's a long enough period of submersion of the root structure, eventually the plant doesn't derive its oxygen actually from the atmosphere, but from the roots. Eventually, that plant will die, right? So I'm trying to use that metaphorically to describe how drugs can have a similar effect on the central nervous system. They are exogenous compounds when they are indeed. They're not endogenous. We're talking about drugs that are consumed outside from the body. And depending on the concentration and the chronicity of the use of those drugs, they may well have a negative impact on a structure in the body, such as the central nervous system. So that was the metaphor that I was going for there. Okay, enough of that. Again, that's me. I am not a MD. I have a PhD. I also happen to have a Master of Science degree. I'm a scientist. And what I'm going to do is talk to you about the molecularity of cannabis in particular regions of the central nervous system, that is the brain. All right, now the paper we're going to be looking at today is this one here out of a journal called Synapse. Will be published, I guess, tomorrow, because today is the 28th of February, although, of course, you can get it early, and that's how I got it. Okay, now, let's just go through some of the reason that this paper is published, some of the background. This will have some social and political timber to it, but basically that's the end of what we'll talk about today on that level. So we know that there is a legalization of marijuana for both medical and recreational use in the United States. And we also know as scientists, and probably everyone else knows this as well, and if you don't, now I can disabuse you of that error in understanding, that legalization of marijuana has actually run quite ahead of scientific, critical evidence-based research on the effects of the cannabinoids, that's the compounds in marijuana, on neurochemical and neuropsychiatric markers. Okay, so unlike other aspects of drugs that are heavily tested before they're marketed as, say, pharmaceuticals or even as some nutraceuticals, we don't know that much about cannabis, the active ingredient, tetrahydrocannabinol, on the central nervous system, even though, of course, marijuana has been around for a long time and ingested by humans probably for millennia. Now, in particular, research has not focused on the central nervous system structures where the cannabinoid known as CB1 and CB2 receptors are abundant. Because in order for the drug to have an effect in the body, it has to bind to a receptor, a protein receptor on the surface of certain cells. Now, those regions of the brain that we're going to be focusing on, and a few more actually, will be the prefrontal cortex, the hippocampus, the amygdala, the striatum, and the cerebellum. Those are some of the major regions of the brain that are going to be impacted by cannabinoids. Some research has shown that chronic cannabinoid ingestion, either smoking or eating, is linked to significant loss of gray and or white matter in the brain. But confirmation of these findings has not been completely forthcoming. That is, there is a fair amount of disparate literature out there. And what we're trying to do now is get away from some of the more socially inclined papers that are published on this subject and try to just do very basic biochemistry, cell biology, cell physiology, neuroscience. That's what I'm going to try to do. All right. Now, what is the breadth of this legalization? I did want to put this up for my own edification because I wasn't really sure. I don't pay much attention to this. Apparently, as of the end of November of 2017, so that would be about 3 months ago, I guess, because this is February 28, 2018, I'm giving this talk, there were 29 legal medical marijuana states, plus DC, District of Columbia, and there were only 9 legal recreational marijuana states. I guess Alaska's one, and of course the entire Western Seaboard, Washington, Oregon, California, Nevada, Colorado, which might have been one of the first states to legalize it, and a couple of states back east, Maine, Vermont, and Massachusetts, for example. Okay, so we know that this is a drug that's being, as we speak, more and more considered for global legalization, and eventually, even though there are federal laws against its use and distribution, there nevertheless are many states now where that is legal. And so if the trend continues, we don't have any idea of whether or not it will. Could be that this drug is legal and becomes as common as something like ethanol, that is alcohol. Hard to say. All right. Now, before we get started on the paper itself, I want to give you a snapshot of the RegioSpecificity of some of the neuropsychiatric elements of the brain. Okay, so that's what's important here for you. You need to get on about the particular nomenclature. So I'm going to put on my glasses here, my visual aids, and go through this with you rather abundantly rapidly. Okay, so first of all, I want to say there are structural and functional associations. Always in biochemistry, we talk structure function. In fact, everything in epistemology is associated with structure function. So there's a structural function association of neural development, in particular in the adolescent brain, both in animal models and in humans, that links cognitive behavioral therapy to a decrease in the stimulation of a glucocorticoid pathway that would include cortisol after stress response. That involves various regions of the brain, the amygdala and the prefrontal cortex, as associated with the hypothalamic pituitary adrenal axis, shown here, as induced by psychological stress. So let's go through this rather quickly because there's a couple of other brain regions we need to think about. First of all, we can discuss things like habit learning and reward learning, and this has an impact on this system. In fact, that's probably how this system evolved, as a reward pathway and as a learning pathway, which hopefully should be coincidental in most people. You feel good when you learn things, like after you listen to a VerEvMed lecture from me, hopefully. That's just stimulation. Stimulation can go from the amygdala to the striatum and from the prefrontal cortex to the striatum in multiple pathways. That's why you see more than a couple of arrows here. It's the amygdala, if you want to look at it very simply, is part of the limbic system. The limbic system is associated with emotion or affect. So what this is showing you is that there is a valence response of all things that are related to working memory, memory of memory or meta-memory, memory strategies, perspective memory, that is what you want to remember, like what I'm talking about right now, in the dorsal region of the prefrontal cortex, as well as in the ventral, semantic memory, extinction learning, conceptual priming, and autobiographical retrieval. Autobiographical, of course, meaning, let's see, when you wake up in the morning, who am I? What's my name? Where do I live? What are my kids' names? That kind of thing, right? So all of that is involved in prefrontal cortex, or so-called the executive branch of the brain, right? Now, you get a constant conditioned emotional learning. That's the point that the psychologists, psychiatrists want to emphasize. And that's what we found by doing these kinds of studies at the molecular level, that the amygdala is constantly interacting via neural networking with the prefrontal cortex and with the striatum. So there's always an emotional nuance to all experience. And I think we all know this intuitively, but we also know it subatomically, subanatomically, and of course at the biochemical, pathophysiological, and normal physiological levels. Now, besides this core system here, we also have the memory system, which includes these regions of the brain, the hippocampus, which includes the entorhinal cortex and the perirhinal cortex. You probably have heard the hippocampus has to do with memory, right? And that's one of the regions of the brain that seems to be possibly negatively impacted in some neurodegenerative diseases, you know, the kind I'm talking about. Now, what kind of memory are we talking about? Declarative, the consolidation of memory, which probably happens during the sleep mode, contextual fear memory, that is why you are afraid when you see the structure of the silhouette of a bear or a mountain lion, or maybe even a figure of a human late at night in the alley. And also complex conditioning. That is all the possible interactions that include the HPA axis, which is basically a neurohormonal axis. So it's a response to all of this learning and understanding that goes on in terms of signal transduction and action potentials being received between one neuron and another at the neuronal synapse. So the HPA axis responds to that and sends out hormones like glucocorticoids, for example. The cerebellum, which deals with reflexive conditioning, such as, oh, this is hot, this hurts when I'm walking on this hurts when I get too close to it, this feels good when I get close to it. Motor learning, that includes things like shooting pool. Like, how do you get better at shooting pool? Well, by practicing shooting pool, right? In fact, we have a billiards table now at home, and we've been learning that is definitely the case. The more you practice, the better you get at playing 8-ball, 9-ball, or rotation, for example, especially if you call it pocket. Their luck is disintegrated. Now, also the sensory neocortex, right? This is everything is involved in the sensoria. Memory storage is also associated with that. Conceptual priming, perceptual priming. That is, right before you see something or smell something, you have a priming mechanism. Now, what I'm trying to tell you is all of this part of the brain is going to be impacted by any exogenous substances you take in, obviously. Now, everyone knows that compounds of interest that we're talking about today, such as THC, or I'm going to talk also about ethanol, we know that if these drugs didn't have a tremendous effect on our consciousness, they would not be recreational drugs, right? People don't drink alcohol, as much as I've heard people say, for their health, right? Nor do they use recreational drugs like marijuana because of some health concern. By and large, these are drugs that make people feel quite different, consciously different, cognitively different. It alters the state of mind. Now, if you think about it, there's my use of that term again, thinking about something when we're thinking about the thinking pathways, which is all meta-analysis, of course, or meta-analysis. But anyways, what we're doing here is we're trying to emphasize, what I am trying to emphasize, not we, the royal we, is that if these drugs have a tremendous effect on motor skills and on learning and on memory and on how you feel about things that are happening to you, you can be sure they have a tremendous effect at the molecular level. So to think that some drugs have very little effect on health when they have a tremendous effect on your conscious bearing is just a little bit over the top ridiculous, right? So they do, and that's why they're drugs that are used, right? Okay, so let's go into more of this discussion. Now, there's all kinds of theories and models that are worked out of how the brain works. I'm not going to go through hardly any of that today. I'm going to give you some basics about neuroscience. Many of you have taken a neuroscience class and psychology classes as an undergraduate, or maybe graduate students, or certainly if you've gone on to medical degrees or PhDs or vet science degrees, or on and on, even maybe some lawyers have learned this. So I'm just giving you very basic stuff. Basically, we talk about a top-down guidance of attention and thought coming from the prefrontal cortex, which can be subdivided here from the dorsal to the ventral, and to the rest of the brain, right downstream. All the sensory cortices, the so-called, as I said, limbic system, the basal ganglia, the hypothalamus, the amygdala, all those center regions of the brain, and of course down here at the arousal center, the cerebellum. Okay, so top-down guidance, which goes to all the sensory cortex, the basal ganglia, which processes the hypothalamus, which includes an association with the reward system, and then the amygdala, which again is a core affect subcortical region of the brain. That's where it is well described, the location, or at least the center of networking, because all of this is interacting all the time. So it's not just the place where emotions come from. That's ludicrously way too simplified. But the amygdala seems to be the place where the emotional nuances of all of the living experience are brought to, reassociated, recombined, and sent back out to the prefrontal cortex so that you actually consciously experience them and reflect upon them over time. So this came from, you see this paper, 2012, from Academy of Child and Adolescent Psychiatry. And what do we want to say here? Look at this. The prefrontal cortex regulates attention, behavior, and emotion through this extensive networking, okay? So the dorsal regions are in blue, and they subserve higher cognitive functions, such as understanding what I'm talking about today. Language is a higher cognitive function. They regulate top-down attention through extensive projections to all the posterior cortical regions we just looked at. In contrast, the ventromedial prefrontal cortex regulates emotion through extensive projections to subcortical areas such as the amygdala. See here, regulates emotion, those red regions there. Red color causes a reaction. That's why they put these in red, right? So now the amygdala, but the nucleus accumbens and the brainstem. In fact, the nucleus accumbens is going to come up in the paper we're going to talk about. The right inferior frontal cortex, or the IFC, is specialized for the inhibition of inappropriate motor responses, by the way, such as if you suffer from Tourette's syndrome, which apparently some people did in history and some people do now. So that also includes inappropriate motor responses that you're not in control of, such as tardive dyskinesia. That is a jerking motion or a shaking motion. That kind of response, right? The IFC apparently controls the tremor response in that way, but also things associated with it, which could be neurocognitive. So in general, the prefrontal cortex is positioned to control all aspects of behavior. So why am I bringing this up? Because the prefrontal cortex is front and center, of course it is, in this talk today. Oh yes. Now, I want to also embed in this a couple of slides of a previous talk we did in VerEvMed on major depressive disorder. Now, why that now? It's because I want you to, of course, reflect on the fact that if we talk about the use of marijuana and the active ingredient THC, we're talking about a population of people in a given country like the U.S. We know that this population of the U.S., there are a fair number of people who are diagnosed with various aspects of neuropsychiatric disorders, including those that involve depression. So I want to remind you about major depressive disorder and know that that discussion needs to be somewhere embedded in our understanding when we're doing research, for example, on how do people with depressive disorders react to, say, the active ingredients in THC, or is it the synthetics for that compound, or the endogenous cannabinoids, in controlled experiments? Because it's a different subpopulation, but it's a big one. Okay, so, sorry, these glasses are bothering me. I'm taking them off. Okay, so major depressive disorder abnormalities have been reported, oh wow, is this surprising, in the prefrontal cortex, the basal ganglia, the hippocampus, thalamus, cerebellum, and indeed in the temporal lobe. In individuals suffering from unipolar depression, the most reliable findings are in the prefrontal cortex, the ganglia, and hippocampus and cerebellum. There's also a possibility of a relationship between the hippocampal volumetric reduction, which is observed in some people with MDD, and alterations of the HPA axis, for example, the secretion of cortisol, the stress hormone. And of course, that's all related or correlated to what's known as the glutamatergic theory of MDD, something I talked about in that previous talk. I'm not going to talk about it any more than that today. If you want to go back and look at that talk, it's all published on my Facebook as a VerEvMed video lecture on YouTube. So let's continue one more slide about MDD and we'll move on. Familial cases of unipolar and bipolar depression show an abnormality in the prefrontal cortex, okay, structural abnormality. Some studies have found altered volumetric measurements, which is okay, does that really correlate? Possibly, but there are volumetric measurements in the prefrontal cortex. For example, a reduction in the amount of tissue found in that system of young unipolar depressed patients, and a reduction in the size of a particular region of the medial prefrontal cortex in adult patients. Post-mortem studies, where we get a lot of this data from, and I'm going to argue later might be a useful thing to do when discussing the effects of THC on the brain. Post-mortem studies have found a reduction in the number of glial cells. Now remember, glia are the non-neuronal cells in the central nervous system. Basically, these are the cells that are associated with the neurogenesis, the transport and the relocation of neurons into specific nuclei. And basically, the whole subanatomical structure of the brain is linked to glial cell modal movement and migration of axons along a fine tangential line associating into networks that ultimately ends up being the networked brain. So glial cells are really important. They're just not neurons. And among the glial cells, of course, you've got microglia, and those are resident immune cells in the brain. And there are a lot of them, and they're going to come up important here in a little while. So you also have glial cells in that same region. So not just neurons. Prefrontal cortex has extensive connections, of course, as we just saw with the amygdala, the nucleus accumbens, hypothalamus. And here are some of the players: norepinephrine, serotonin, and dopamine. And these have their own systems, their own neuronal complexes, etc. And again, I'm sure many of you have heard about this. Most of you health professionals know a lot about this. And certainly, if you want to go back and review that, you can go and visit some of my video lectures, or just go back and consult any given textbook and you can look at that. So functional imaging has become popular, and we know that functional imaging studies have reported these abnormalities in function and structure. Increased activity may occur in some areas of the PFC, particularly the posterior orbital, the amygdala, and in the anterior cingulate. Consistent with the literature that suggests that sadness and anxiety in normal subjects increases the activity in those areas of the PFC, okay? And the increases in the ventral, orbital, and cingulate cortices may be related to emotions and or obsessive ruminations. This is all in the psychology, psychiatric literature. I'm just reminding you that this is embedded in this talk. All right. Now, back to the paper in Synapse. Love the name of that journal. Psychoactive drugs alter the structure of neurons in the prefrontal cortex and the nucleus accumbens in animal models. So there's a lot of this work we're going to talk about today is in animal models, and that's because of ethics, and also because not just because of ethics, but also because we understand the animal neurochemistry much better than we understand the human neurochemistry. Thank you. Now, THC injections, which I don't think is the normal way that people take in THC, but okay. I think they normally smoke it, or I guess they digest it. I don't know, but that's what I'm told. THC injections in lab animals correlate with alterations in dendritic arborization in prefrontal cortex, striatum, hippocampus, and in the ventral tegmental dopaminergic neurons. I know it's a mouthful, but all we're talking about here are those subanatomical regions in the brain. Once you've already seen a couple pictures of it, don't worry, you'll see more. Now, this is what we mean by arborization, this kind of branching. Remember, arbor comes from the tree metaphor that I started my talk with, right? So arborization just means a branching mechanism. These are going to be dendrites. Remember, the dendrites are all going to subtend into a synapse in the adjacent new neuron, right? And so that's how signaling occurs. The action potentials are going to cross through that synaptic cleft from these endpoint butons of the neurites, or that is the spike shafts of these arborated or arborized structures into the effacing synaptic cleft into the next neuron, right? So dendritic branches arborize, again, which is called further branching, and thus generate plasticity. Now, neuroplasticity has gotten a lot of prime time discussion in the journals, and probably it's also filtered down into common lay people talk and into magazines. I don't know because I don't follow any of that. But neuroplasticity basically is a new term which people like to say is really important, because plasticity in this sense means that it is malleable, that the central nervous system is malleable. That means you can learn things, right? So the more neuroplastic the brain is, considerably, within a lot of caveat, consider the more healthy brain. So if you have a brain that can learn, it's not neuroplastic. So that's what neuroplasticity means. It means that there are structural and functional alterations of the actual brain chemistry and brain anatomy that relate to learning and understanding and remembering and becoming self, right? So that plasticity is generally considered an important good thing. The more neuroplasticity, some would argue, the better equipped you are to learn new things, and the longer you live a good, cognitive, healthy life. Now, there's some arguments there, here and there about that, because obviously too much neuroplasticity has been linked to things like massive confusion, right? As you might guess, too many inputs at once, for example. And that's just a good way for me to segue into the further of this discussion. So bursts of neurotransmitter from a nearby neuron, adjacent neuron, land on the dendrites receptors and trigger an electrical wave, and that's called the dendritic spike. If the local dendritic spike is powerful enough, it can propagate to the main body of the neuron, generating an action potential output or a nerve impulse, we can call it. Arborization, shown here, enhances this, so alteration in arborization would corrupt action potential firing. Action potential firing is the main game in the brain, okay? If you don't have action potential firing, you don't get any transmission of any neural circuit. So obviously arborization is going to affect that because of this basically increasing in surface area and therefore quickening of the response, and also it's believed an elaboration of the signal. So you don't just get something like blue patch now, you get blue eyes in a face against a black backdrop talking to you some strange language of neurochemistry, right? So all the nuances that you get besides just looking at the very basic facts, right, is elaborated because perhaps, first of all, neuroplasticity, but also maybe the level or the discrete kind of arborization, right? So it's a dynamic model. That's the important thing. This is a dynamic event horizon ontology. It's not a substance ontology. You don't just have the brain and there it is, and somehow it gets filled up with thoughts and ideas. You get old and then you die. It's a constant changing system, constantly changing system. So it has an event ontology, an event reality in terms of metaphysics. Okay, so here's a paper. Here's a slide I got from a paper called from Genes and Development 2010. And it's just showing you some of the fine points of a neuron, right? So here's the dendritic arbor out here. And here's some of the things it does at the molecular level. Now we're looking at this. Here's the presynaptic terminal. There's the postsynaptic terminal. You have two neurons that are interacting here, right? There's the dendritic edge right there, and there is the synaptic cleft between the two, right? All right, so that's what we're looking at here. We're blowing that up, blowing that up. The dendritic arbor here, what does it do at the molecular level? It's involved in RNA transport, delivery, and what? Protein synthesis or translation. Take the RNA and you synthesize the protein there. So if you think about your neurotransmitters, the peptide neurotransmitters, are they sent along the fine end of this entire axon as a protein? No. They're actually, the RNA is sent. It's kind of an unusual thing. The RNA is processed, that is, it is prepared for translation, and the protein is synthesized de novo out here in the arbor. You also get microRNA de-repression, and you also get, and also this is going to be associated with any number of synapses. So 10 to the 15 total synapses perhaps in the adult brain. That is a big number. All right. Now, as you move away from the dendritic region, you go to the cell body, or soma, which means body, and that's where you get our alternative splicing of that messenger RNA, RNA packaging, and microRNA silencing. Remember, microRNAs are RNAs that are single-stranded, and they bind to messenger RNA, it's the most simplistic case, and they basically prevent it from being translated. So if there's sequence homology between a microRNA, a small interfering microRNA, with a messenger RNA, sufficient nucleotide sequence compatibility, sequence similarity, that microRNA will bind, it will prevent translation because you can't translate a double-stranded messenger RNA. It doesn't happen. Okay, so that's why this is all over this paper here. That's what they were talking about in this paper, apparently. There's your axonal shaft. That's the longest piece of the neuron. And, of course, that's where you get transport. And, of course, you can also get microRNA silencing of the message there. If the binding of the messenger RNA occurs there, you're never going to get translation out here in the dendrite. And then there's the growth cone, okay, that's going to guide this whole process. There's RNA delivery and translation, and microRNA, again, de-repression. So there's a lot of mimicry and overlap within the neuron, but you can also see there's a lot of division of labor. So what does this say? Post-transcriptional mechanisms like alternative splicing. If you don't know what that is, go back to one of my previous talks. It basically means taking a nascent RNA and splicing up the exons and introns in such a way as to making splice variants, which when fused together and then translated into proteins will give you different proteins because they're going to have different exons fused together. Those proteins will then have subtle different functions, yes, maybe having still a core function, such as if it's an enzyme, for example, or some cytoskeletal structure. So that alternative splicing in 10 seconds. RNA transport and, again, interfering RNA silencing. Alternative splicing occurs in the nucleus of the cell body, too. It's important for generating protein isoforms, and that's what I just meant by that. Following splicing and mRNA maturation, that involves the capping and the polyadenylation and removal of that, and, of course, all the splicing. The transcript can be packaged into transport ribonuclear proteins, which is really important in the neuron, and it can be repressed by ribonucleotide binding proteins. So we can retard the movement of that RNA, or indeed, as I just said, interfering RNAs, delivered to distal sites of the neuron, and locally, these small RNAs, interfering RNAs, involved in regulating protein levels throughout the cell. So that's a really important thing to keep in mind. There's a lot of interfering RNA out there, microRNA, which controls gene expression, and this is post-transcriptionally. All right. Now, I like this slide. I got it from some Commons site. I just want to show you some of the different possible, these are just like, skeletal drawings of what's seen under probably a microscope of various kinds of arborization. You get all kinds of unusual structures here. The body can be at the core, and you get branching out. You get branching that's only lateral diffusion, is only branching in one direction. It's not along the entire central axis. You can have more diffuse branching, bigger differences between, or spaces between each of where those boutons will end up. You can have an inverted cell body or an extroverted cell body. You can have multiple branching mechanisms like this, a proliferation of branching. You can have this. This is my favorite type. This is just like overall branching within a branching within a branching, and all these other like conical shapes, etc. What I want to say about these is specific patterns are still being analyzed for functional differences. You see these patterns, it's a structure, it's a macro structure, so you'd guess there's some function to it. Whenever you see a structure, think function. That's like paradigmatic epistemology, paradigmatic actually biochemistry. Density fields help elaborate surface area. You get more surface area when you cause more elaboration of the branching, and thus alter receptor density, because remember, all this branching has receptors on the surface. What kind of receptors? For example, receptors for the neurotransmitters. What else? Receptors for things like, that's right, THC. Okay. Anyways, all of that arborization that you get drives itself toward a potentiation that leads to memory, cognition, and deficits therein, right? So the more diversified these arborizations occur, the more elaborated they are through some spatial temporal awareness from a previous event ontology. The sophistication of that branching mechanism is ultimately going to lead to different levels and quality of memory, quantity and quality of memory, and also sophistication of cognition. Okay, this has been determined again and again in animal models, running through mazes and complex environments, but also with humans, and particularly a lot of aspects of looking at brain post-mortem. Now, what we already know from the literature, okay, so this isn't any kind of political statement at all or anything else, just THC corrupts this process. We know this because all kinds of things corrupt this process that people ingest. Even a high-fat diet can corrupt this process. So to say that THC does is nothing heretical or nothing to do with any kind of statement other than saying, especially purified forms of psychoactive drugs, as you might guess, psychoactive, it works in the brain, it's going to have a much more complex and determined potent effect on things like arborization than, for example, high-fat diet. High-fat diet may have an effect, but it's not going to nearly have the effect of a highly potent, purified substance you take in the body exogenously. As we're going to find out, THC is not the only player. Stimulant drugs like cocaine and amphetamine also alter the arborization, as does ethanol, which I called another drug. Now, current paper here, we're going to go through pretty quickly, addresses the potential for tetrahydrocannabinol to alter experience-linked neuronal plasticity. Remember, that's what it's all about. Experiences are going to cause an elaboration of that neuroplasticity. Experiences like learning, like right now what you're doing. A learning experience that is known to produce largest effects on neuron structure results from, this is the animal model we're looking at, housing animals in complex environments. So you take an animal that was in a cage and you put him in like a little animal house world where there's like multiple places for the animal to move around in, a lot of room, a lot of places to hide if they're a rodent, for example. That is going to alter the structure of all of those anatomical regions in the brain. This has been known since the 50s. Even before that, in the 30s and 40s, when staining was done on neurons, all the early staining work showed this, okay? You get more complex arborization when you have a more complex environment. That's how it works. Furthermore, the administration of psychomotor stimulant drugs to rats, again, amphetamine, cocaine, nicotine is psychostimulant, alters the ability of the later experience in a complex environment to change dendritic structures, sometimes occluding such effects. So if you get drugs and you put an animal into a complex environment, there are sometimes what happens is that the drugs diminish the amount of arborization and learning, for example, in a complex maze, in animals that have been previously exposed with the drug. That means that prior arborization and all the neurochemical nuances besides that, massive general structural alteration, are going to lead to changes in the ability for that system, that stratum now, to react to a complex environment. That means alterations in behavior that can be long-term, in fact, the life of the animal. That's what's important here, right? Complex things that are permanent, or at least appear to be semi-permanent in a rodent's life, and maybe, who knows, in higher animals such as primates like man. Now, this paper is going to discuss whether THC might alter brain's subsequent response to complex housing. That's what they're doing here. Previously, it was demonstrated by them and other researchers that THC effects are limited to the prefrontal cortex, and the orbitofrontal cortex, as well as the nucleus accumbens. Now, those are main regions they're looking at. I can tell you those aren't the only places where THC has an effect, because I've looked at the literature, a lot of places. For example, as we're going to see soon, in the microglia of the brain, which aren't neurons at all. All right. This paper examines the effects of THC on spine density. That's part of this whole arborization. And what they predicted was that dendritic spine changes, including branching and length, might both respond to complex housing and THC, both, okay? Because those are two different, basically, treatments. Also predicted that psychomotor stimulants, because THC is like that, or they're assuming it's like that because it's a neuroactive drug, a neuropsychiatric drug, works not that differently, surprisingly, from a lot of other drugs, including drugs like ethanol. When you finally get down to it, it's a reward pathway. THC could also alter morphological changes related to complex housing. That's what one of their hypotheses was. And basically they tell you, yeah, it looks like they confirm this. Okay, so let's take a look. Okay, first of all, very, 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 very simple paper, very simple structure of the experiments. They gave these rats either 0.5 milligram per kilogram of tetrahydrocannabinol purified to delta-9 isomer or saline intraperitoneally daily for 11 days. So it's a short experiment. Remember, these are rats. The dose of THC they claim was selected because it was in the estimated range that humans might use. Okay, I guess. I don't know. I haven't done the calculation, but okay, I'll take their word for it because these are people who study this. All right, and recreational use, not medical use. Now, what first thing they did is they just want to say, well, does THC have any effect on like locomotor activity, like movement around? And it appeared to have a significant effect. You get increased activity of these animals that are given THC versus animals that are given just regular phosphate buffered saline. They seem to be more active. Okay, that's interesting because one of the indirect things you might be looking at here is the fact that that kind of movement around is going to alter, again, those subanatomical structures in the brain. Just moving around, more experience is going to do that. So that's probably why they did the experiment, but still sort of an interesting thing. But you see towards the end, they were seeing less and less of that. Of course, that's because, it's the end of the 11th day, but who knows what happened after that? I don't know. All right. Here's basically all the data we need to worry about. And what it says is dendritic branching, but not length, were affected by complex housing, and there was an increase in spine density and in the complex housed rats. So complex housed rats were different than THC alone. THC increased dendritic length in both cage and complex reared animals, and branching was increased only in the complex housed group, and the THC increased spine density in the complex housed group, but decreased it in the cage. So there was a environment by treatment effect, okay? Treatment being the THC. So the S stands for significant, the N means not significant, okay, no effect, and the S with an asterisk means the opposite, a significant decrease. So what I point to here is in these are just cell types within this area of the prefrontal cortex. You have a significant decrease in the branching of this cell type in the PFC, whereas you have a significant increase in THC, okay? And if you look at what happens then, because they were exposed to THC, the housing by THC effect, there's no effect because the housing doesn't have an effect anymore because the THC has basically washed that out. So that's what they're already showing. They're showing that having preconditioned with tetrahydrocannabinol can have a negative effect on what normally occurs by complex housing effects on those anatomical structures vis-a-vis the arborization, vis-a-vis cognitive behavioral effects, because these have been causally linked in, probably over 75 years of studies in the animal models. So there's some significance there. Let me put the glasses back on. Here's no effect on the ID length, significant effect from THC. Again, there's the negative for spines and the positive for housing and the positive for THC. These are tremendously opposing effects, and then as a result, there's no effect ultimately on how housing superimposes an admonition of the THC on this arborization of these prefrontal cortical neurons, and on and on. So there's a lot of difference there, but it's all basically summarized right here. So you see a fair amount of differences, and the data showed this. I'm not going to show you all the data, but they actually took measurements of all this, very careful measurements, not just with dyes, but also with densitometer readings and whatnot. So this is good, accurate data. I'm just summarizing it for purposes of this talk. But you see a fair amount of significant differences between the THC and the housing. There is some interaction between housing and THC, such as the branching of those neurons. Let's see, the branching of the nucleus accumbens here, even though this has significant effect and this did not. Remember, housing then, if there's no effect with THC, housing does have an effect. You're going to have a significant effect when you do the pairwise comparison. So what are the conclusions of this paper? And we're not done yet, but I'm concluding this paper early because I want to get to the rest of some of the literature. Paper is very simple, and it answered some very simple questions. So they weren't trying to rewrite the textbook on neurochemistry or THC effects on neurochemistry. They were only looking at a few things here. Conclusions are, THC alters both dendritic arborization and spine density in prefrontal cortex and the nucleus accumbens in the rat. Like psychomotor stimulants, THC interacts with the effects of complex housing, you saw that, on neuronal morphology, and again, it was both in the prefrontal cortex and in the nucleus accumbens, important places, as you know, in terms of behavior and cognition in the animal. And so they say the effects of complex housing vary with cerebral region, okay? No argument there. Let's do the dialectic now for this paper. Class is off again. Thank you. It's clear that psychoactive drugs can limit or alter the effects of later experiences. That was the housing and drug interaction. In modifying neuronal circuits. And it's possible that one long-term consequence of psychoactive drug exposure like THC may be an alteration in behavior and psychological function because we know they are related to the alteration of arborization in those regions of the brain. Okay? A potential mechanism for those effects, okay, you want to know mechanism, at least I do as a biochemist, are, of course, all those things we talked about at the beginning of the talk. Transcription, splicing, interfering RNA interactions, as well as perhaps messenger RNA turnover via the ribonuclear protein complex, and of course the transport itself. Those are some of the places, the molecular locations where these alterations could be occurring, right, that end up with changes in arborization, you see. Perhaps early, chronic, and later adult use of psychoactive drugs can induce a neuroplasticity that would otherwise naturally occur during reward circuitry, that is, doing things and feeling good about doing good things, which is how your brain is wired. You feel good when you do good things. You feel good when you hold a baby. You feel good when you smile. You feel good when you do nice things for yourself and other people. These are natural rewards. You should feel good about those things, right? But they're saying that normal development upon appropriate stimulation may be mimicked by the effects of the drug. Now, that already should raise a red flag. That means that the drug could be substituting, depending on when it occurs temporally, when generating these reward pathways, in, for example, the adolescent brain, or in utero in the gestating fetus, as we know from alcohol, fetal alcohol syndrome, which I'm going to get to in a minute. Studies on THC for the gestating human have not been done to my knowledge, and so they definitely need to be done. But let's move on. THC-induced reawakened, these are my terms, developmental mechanisms, because the mechanisms that are normally happening during the temporal development of the brain, reawakening them, altering them, sending them in a different direction because of the drug, could form unusually strong, that is emotional as well as cognitive, long-lasting, and maybe maladaptive changes in the brain. Maladaptive only because they're not the normal progression of events. For example, the reward pathway. Altering reward pathway by rewarding yourself when there's nothing directly in terms of an event ontology that should feel rewarding. In other words, an artificial reward like what the effect of the drug is on the person's consciousness, right? Alcohol, for example, or marijuana. So clearly, if this is all something that we can digest here, this would be dependent upon the density and turnover of the receptors, because THC is going to function. You have to know the density of the receptors, CB1, CB2 receptors. And of course, the turnover of the ligand, that is the endogenous ligand. Remember, we have endogenous cannabinoids. This is in place not for people to smoke pot and affect it, right? You have endogenous cannabinoids, and that's why we have receptors for it. But what about the pharmacokinetics of the digested drug, right? What about the metabolism of the drug? What about the pharmacodynamics of the drug, right? What about both of those things and how the drug is interacting with the entire system and whether or not it's acting all the time at the same time in certain regions of the body, particularly in the central nervous system? Now, another thing I bring up that they don't is that epigenetics would also play a critical role in arborization and the extent of density increase or decline depending on the stimulus. Now, we already know this. We already know that epigenetics alters gene expression. So what we just talked about, this whole arborization process, RNA transcription, RNA splicing, RNA processing, ribonuclear proteins, ribonuclear binding proteins, translocation across the exon axis, and also, of course, the translation itself of the polypeptide and the utilization of the protein. All of that can be epigenetically modified, which means environment plays a role. So normally the environment's doing things like that, impacting on branching and neuronal circuitry. Of course it is, because that's a part of adaptation. Of course, learning is probably largely an epigenetic phenomenon, although I can't argue that I know it's the most significant thing. We know that the alteration of the environment plays a large role in gene expression, though. That's clear in animal models and humans and in bacteria and even in viruses and even in plants. But the extent of that we don't know, and the extent of what drugs do to that system is the real unknown, and that's the point. Okay. Now, let's look at a couple of other papers. Paper in Neuroscience, again published very recently, 2018. You see that. This is not looking at THC, it's looking at ethanol. Okay. Ethanol abuse during pregnancy may lead to something we all have heard of, fetal alcohol syndrome, or also larger known as the spectrum, fetal alcohol spectrum disorders, or FASDs. They involve, we are clear to understand in humans, structural brain malformations with, of course, accompanying very unfortunate cognitive and behavioral deficits. So fetal alcohol syndrome is a bad thing to happen in women who are pregnant, who drink enough alcohol that causes to their infants, definitely have a destructive effect on the entire life of that child and that adult. Neuroplasticity and connectivity are affected by in utero alcohol exposure. There's no doubt about it. So if you look at the specific thing that's going on, neuronal plasticity is corrupted as well as connectivity. So there again, we're talking about arborization. In fact, in the third trimester in mammals, this is rat here as a study, it's marked by massive increases in brain size. It's the same actually that happens in primates. Proliferation of, although the brain size increases after gestation, after parturition actually in humans. Proliferation of astrocytes and oligodendrocytes, those are the glia, and dendritic arborization. So all of that is a tremendous developmental thing that occurs in the third trimester. That would be the worst time to have this ethanol there. Ethanol exposure in that third trimester, therefore, is linked to microcephaly, cerebellar and hippocampal abnormalities, hippocampal and prefrontal cortex, apoptotic neurodegeneration. Very bad stuff to the brain. Ethanol also alters hippocampus-dependent behaviors in several rodent models of this complex syndrome. Okay, so we know this, and this neuroscience paper was discussing it. Now, continuing a little bit more, choline, which is actually trimethylethanolamine, and it's actually a head group on major phospholipids known as phosphatidylcholine, and what's the other big one? That's correct, sphingomyelin. Choline is going to be that head group on both of those lipids, those membrane lipids. Anyways, choline by itself improves hippocampal functions in the adult and aging brain, maybe because of its effect on the membrane. But it's also metabolized in a way that generates neuroactive substances. Choline can prevent hippocampus-associated behaviors in rats exposed to ethanol during brain development. Wow, that's interesting, right? Ethanol causes long-lasting changes in dendritic arborization, as we've already alluded to. Neonatal ethanol exposure decreases spine density. Here we go again. Dendritic complexity of basal dendrites as well as dendritic spine density in apical dendrites of layers 2 and 3 of pyramidal neurons of the medial prefrontal cortex. And that can all be reversed. Look at this, amazing, by voluntary exercise in these rat models. So remember I told you environment could play a major role here. So we don't just want to think about the ingestion of these toxins like ethanol. We also know that there's a tremendous effect just on the lifestyle and whether or not that has an effect on the alteration of brain structures, even when that brain structure has been negatively impacted by ethanol ingestion during gestation. Right. Okay. So what are take-homes that I get from this paper? Real briefly. Alterations in structural plasticity and neuronal cytoarchitecture, such as dendritic arborization and neuronal morphology in the PFC of adult animals, were associated with that synaptic plasticity and behavior. Okay? Ethanol increased the arborization of apical dendrites without affecting the reach toward the hippocampal fissure, so they didn't alter length. Now, that's something we saw that looks very similar to the THC study in the rat model we just looked at. Yeah, that's why it caught my attention. Two papers didn't, of course, reference one another because they were published at the same time, which was like right now, 2018. But that's interesting, right? Yes, it is. It's interesting. So, developmental alcohol exposure induces a premature maturation, because that's basically what happens, because it's a termination of these dendrites, because they stop, the length stops, they just branch, of apical dendrites in hippocampal pyramidal neurons, which could lead to premature restriction in neuroplasticity, because you don't have then necessarily generating new synapses, because you have this constant branching. And I didn't mention it before, but I will now. Excessive arborization is the reciprocal of synaptogenesis. Synaptogenesis, then, again, is generating the synapse to be able to bind to the boutons to the dendritic branch. Excessive dendritic branching is going to overload the system, so you're not going to be able to generate new synapses. So synaptogenesis is not afforded by the increase in arborization. So they're not mutually exclusive, but they have a reciprocal relationship when you get to the high end of the quantity of one over the other. And that's not a good thing, because a lack of synapses with all those extra dendrites, one of the things that can happen there, as we're going to see in a moment, is that can induce an inflammatory response. And when it's in the central nervous system, that can be quite damaging, as you might guess. Okay. Now, it's another paper. This is the European Journal of Neuropsychopharmacology, published just exactly 2 years ago. And look what this paper is about. Back to cannabinoid. Chronic cannabinoid exposure during adolescence induces long-lasting changes, including 1, significant altered dendritic arborization of pyramidal neurons in 2 and 3. Didn't we just see that with alcohol? Yeah. Impaired hippocampal input induced synaptic plasticity in the PFC. Again, we saw that in ethanol. Remember, the length of the axon shortened. It doesn't make it all the way to the hippocampus. Significant changes in the expression. Now, here's something different. Now I'm starting to talk about the individual genes and their proteins. A protein known as PSD95, which is postsynaptic density protein, 95 kilodaltons, and that is a membrane-associated guanylate kinase. So it's part of the MAGOC family of proteins. I've mentioned this before. Sorry for that horrible acronym, but there it is. I didn't make it up. And what that normally does, that protein, is it heteromultimerizes, means it makes that heteromultimer, right? And when it does that, it's recruited into some much larger protein complex called the NMDA receptor. Okay. And that in association on the membrane in the neuron with potassium channel clusters. Now that, in other words, you're making a megaprotein family unit, right, by this one change in the expression of the PSD95 protein. which seems to be altered from THC ingestion. So all of that then may form a multimeric scaffold for the clustering of receptors, ion channels, associated signaling proteins in the medial prefrontal cortex. Now, if there is an alteration in expression of that, what's going to happen to all of that clustering, all that protein-protein interaction? If nothing else, it's going to be corrupted. Negatively, positively? Well, who knows? Certainly it's not going to have the right stoichiometry. If you get an alteration in specific expression of specific genes that are members or subunits of that larger mega protein complex that has to still be embedded in the membrane. These changes, therefore, in synaptic structure and function in the prefrontal cortex may correlate with long-term cognitive deficits induced by adolescent cannabinoid exposure. This is the important point here. Cannabinoids may impede the structural maturation of neuronal circuits in the PFC, which is what it looked like it did here, thus leading to potentially impaired cognitive function in adulthood. Okay? Remember, you're getting developmental, neurochemical, subatomical alterations during the early stages of adolescence, right? That's when this is happening, all the way till you're about 20, 21, when you get a whole lot of dendritic pruning back. And when that happens, that's when the adult brain starts to rear its head. And that's when all the changes that are happening normally associated with late puberty tend to start to decrease. Okay, so there is a neural correlate to that behavior, if you ever wondered. Now, paper from Journal of Neuroscience 2014. High-resolution magnetic resonance imaging scans on young adult recreational marijuana, this is humans, and non-using control people, revealed greater gray matter density arborization in the marijuana user than in the control participants, particularly in the left nucleus accumbens. We already talked about that structure. Extending all the way to the subcallosal cortex. That's really important for regions I can't get into right now, but it has where two sides of the brain interact. The hypothalamus, the sublenticular extended amygdala, and the left amygdala. This is all very important because it links emotion with those higher executive orders and this transmission of thought processes throughout the circuitry of the brain. Okay, so that's really important, right? In young recreational marijuana users, structural abnormalities in the gray matter, remember, that's all the neurons and all the dendrites, right? And even the glia. Structural abnormalities in gray area density, volume, and shape of the nucleus accumbens and amygdala can be observed. That's using MRI. This isn't done post-mortem. This is just using MRI, doing scans of those regions of the brain in living adults, or in this case, young adults. These findings suggest further study of marijuana effects are needed to help us inform discussion about the legalization of marijuana. And notice when this was published 4 years ago, a lot less legalization than there is now. Now, we followed up on a lot of this work. I have to say, sadly, no. These results extend prior studies, however, showing that drugs, this is them talking 4 years ago, showing that drugs of abuse that are known to elevate dopaminergic pathways, dopamine release, this is the reward pathway, are associated with structural abnormalities in the brain, and that can be related to disruptions in behavior. There's no caveat here, because obviously if you alter this reward pathway, ultimately you're going to lead in changes in wiring and firing. That's going to give you structural abnormalities, and structural abnormalities in the brain usually leads to some kind of disruption in the output, which could be cognition and behavior. Now, let's continue. A paper published in 2016, The Frontiers of Neuroanatomy, another very glitteringly exciting journal, I can tell you, looks at various subcell types. And one such subcell type, which we're going to find out is in the amygdala, is one that has cannabinoid receptors. Okay, and these are called basket cells for a reason I'll mention in a minute. So CCK, that's cholecystocytokinin, which is always a protein associated with cannabinoid receptor 1, had more higher order dendritic segments, which together were longer than those observed for the other two cell types that they're going to look at, resulting in longer dendritic length and further away from the soma, or the cell body. Now, these are not studies that are done by adding THC, or these are just cell types. So what this tells you right away, full stop, is that there are cell types, these CCK, CB1, BCs, these basket cells, which are really important in a certain region of the amygdala, which by themselves perform or generate a platform where there is a different type of arborization, which will likely be impacted specifically by binding of the ligand, either the endocannabinoid or the exogenous THC, to the receptor in these cells, differentially from other cell types. That's what this shows here. Sorry for all the complexity. Actually not very complicated. We're looking at parvalbumin, exonic exenol cells, and we're looking at these CCK cannabinoid basket cells, 3 different types. And this is just giving you an idea of what that arborization looks like. That's what it looks like. It's different between these cells, more similar to the parvalbumin, but subtly different from these cells here. So you've got parvalbumin, exonic, and cannabinoid basket cells. Now, these basket cells are, by definition, inhibitory, of course, because it's GABA, GABAergic interneurons. Those are the 3 different types of interneurons showing all this arborization. Now, the rest of this data here basically shows this. CB1, basolateral amygdala, GABAergic neurons, these guys here, their arborization is enhanced compared to other subtypes just by endogenous control over presumably endogenous endocannabinoids. And all of this basically just tells you that. Okay, so box chart comparison, total dendritic length. You're looking at then specific measurements here. Comparison dendritic length of the 3 different cell types and alterations thereof. All of this data supports this one general point. Okay, one general point is that those neurons that have the cannabinoid receptors are poised to have an altered arborization that is unique to that subcell type. And there's a difference in length, as you can see here, and also in some of the branching mechanisms. You notice too how it's, rather than starting from the cell body and sort of extending out like as diagonals in an old-fashioned city, you have much more where there's a bifurcation of pathways, much more branching in these preformed cannabinoid receptor amygdala cell lineages. Now, what's the dialectic here? Arborization is a critical developmental neuronal process that is highly specific according to brain region, temporality, that is the developmental process we talked about, and involves density and length. Okay. Inappropriate arborization is caused by THC or ethanol. These are the papers we looked at. Might lead, can lead, looks like it lead, according to this research, to structural abnormalities, and maybe those are linked to behavioral and cognitive deficits. Those all definitely are associated in animal models and in human models. Human studies, excuse me. Some studies have failed to reproduce those effects. Now, that's a really important point. So if you look at the literature, there's a lot actually that's in the literature now that talks about that it's waxing wonderful about synthetic cannabinoids and exogenous THC and even smoked inhaled cannabis products on various types of diseases such as MS, maybe even Alzheimer's and Parkinson's disease and other kinds of neuropsychiatric disorders, with the exception, I think, of major depressive disorder and schizophrenia. I don't think those 2 disorders have been linked positively with chronic marijuana use anyways, probably just the opposite. Now, why is there this difference? Okay. Now, I already told you that there is a very strong bias in the literature that does not have enough data about this whole anatomical difference with drugs and the alteration of arborization and the alteration of specific subcell structures within given subanatomical regions of the reward pathway, as per example that we've been doing. Okay? Therefore, the responses have not been well vetted or described. There's evidence, I just went through some of it, but there's a lot of evidence where they say, well, we can't find that. That doesn't mean, though, because they can't find it, that there's something else there. So what I say here is failure to reproduce data does not mean the premises are invalid. Premises here being the hypothetical deductions that end up becoming the hypotheses. So run the experiments that give you the results that are then looked at as data and analyzed by yours truly and others to do new experiments. Now, why is that? I would argue it's because science is not like my beloved philosophy, which I love almost as much as I love science in terms of something to do with my brain. That's because in philosophy, even though philosophers like to think they have empiric intuitions, and certainly they do in many ways, they're not generally an experimental field, experimental discipline, experimental science, philosophy that is. Although they're starting to dabble with it with psychologists and psychiatrists. So that's a whole other discussion I just want to bracket off right now and say no more about. But they're starting to look, philosophers are starting to look at, what is the philosophy of consciousness, for example. There are courses like that taught in fact, in universities I've taught in. Okay, but it's in its infancy. The point I'm trying to make is that science doesn't have such a strong interest in throwing off paradigmatic laws or principles. So when you get a lot of evidence that says that glycolysis is an anaerobic metabolism of glucose to pyruvic acid in basically every cell that's ever been looked at, and there are some 7 reactions in the pathway, and there are intermediates that help regulate the pathway, such as phosphofructokinase altering the structure that is the synthesis and degradation of bisphosphorylated fructose 1,6-bisphosphate, for example, and how that regulates the pathway, all the stuff that I talk about in terms of metabolism. We're not so easy to just dump that off and say, well, we got new data, we're just going to throw that out. So there's more dogma in science than there is in philosophy, and that's probably a negative thing, but by the same token, it also is reflective of the fact that we generate a lot of data. And data, as many scientists argue, don't lie. Now, I don't think data tells the truth or lies. It's humans subjectifying the information and determining what looks like truths with a small t. The data itself don't have anything to say about it at all. They're just data. And in fact, they're the result of a thought process that involved a hypothetical deduction that led to the experiment, that led to the result, etc., like I just went through. The so-called scientific method. Big, huge italics and inverted question marks on that one. And the question marks include, quotation marks. But there's a question mark as well. Throw that in there too. All right, so what am I saying? I'm saying that when you've got data like we just looked at, and then you have data that doesn't support it, don't throw this data out. We don't have enough information. Don't invalidate the premises just yet. We don't have enough information. That, as a scientist and as a philosopher, I think I can stand behind. Now, I say, my conclusion here, on the contrary, ethanol, THC from marijuana, and other drugs, of course, do cause, and I put that in quotation marks, permanent neuronal damage. They certainly seem to in these animal models. It's more than just like guessing. They do. And that does translate to apoptosis in many cases. And that translates or then results in the animal models for sure, behavioral and cognitive damage and deficits in terms of, say, mazes, and in terms of how the animals interact with how we measure cognition in those animals. Now, here's a couple of things I want to leave you with. This is like the last thing I added. I'm sorry it's going so long, but this is really important. A couple of final concepts and ideas from the review literature, not from the primary literature. 2 papers, Frontiers in Pharmacology, a 2012 paper, and then the journal called Pharmacology and Therapeutics, published just October 2016. That's only a couple years old. Or less, actually. Let's go through this quickly. Neuroplasticity plays a key role in maintaining action potentials by simultaneously establishing new wiring and firing networks. We all agree with this, okay? Arborization, which I've already explained to you, of neuronal dendrites is part of that system, along with synaptogenesis, which we haven't talked much about this afternoon, and a complex molecular machinery that selectively prunes and sculpts the synaptic cleft. Now, the maintenance neural synapse dynamics, what this system is regulated by lots of different enzymatic and lipid machinery. What enzymes? Things like extracellular metalloproteinases, which I know I've talked about many times in cancer. plus cadherin proteins, which I talked about in a review article I wrote both on anger and another review article on autism spectrum disorders. I can give you the citations for that if you're interested. Just email me. And a tight control over neural turnover and neurogenesis. Now, that's a lot to say, but that's basically what we're saying here, is that maintenance neural synapse dynamics has a lot to do with what's going on in real time in terms of enzymatic activity and also the expression of the receptors that can be stimulated by endogenous or exogenous ligand. Now, neuronal remodeling also requires the immune system. And in the CNS, a major player in that network is the resident macrophage known as the microglia. And they can be activated as in neuroinflammatory responses. And we know this. Microglia, resident macrophages, can be turned on. They can cause neuronal damage because they can generate an inflammatory response. This happens in classically neurodegenerative diseases. Okay? Now, microglia, we're not done here. Sorry, sit back down, please. Microglia can exist in at least two species, two isoforms, that are reminiscent of macrophage lineages, of which they are, of course, family members. Type 1 microglia phenotype is a pro-inflammatory and pro-apoptotic type, while the type 2 microglia, again found in the central nervous system, is basically a neuroprotective microglia. That is, it generates not pro-inflammatory, but anti-inflammatory cytokines in some instances. So it's neuroprotective, and that it also includes a protective neurogenesis associated with the type 2 that is helping the hippocampus with the mobilization of new neurons to sites that need new neurons, and then the hooking up of those neurons via the axonal terminals to the adjacent neuron, right? So, and also neuronal repair mechanisms. So type 2 microglia are really important in stopping or maintaining or slowing down the neurodegenerative process, which is likely to occur towards any kind of ischemic or damage to the central nervous system, which is also necessary to get rid of damaged cells, right? Okay. Now, microglia are implicated in the recruitment and integration of nascent neurons into established nuclei, both again after damage, such as you get some kind of ischemic attack like a stroke, and even during healthy maintenance surveillance, because they turn over, right? Dendrite arbor pruning and synaptogenesis and neurotransmitter patency are all part of this. So the arborization at the beginning during, say, juvenile development, the pruning of those dendrites in humans around the ages of, like, say, 19 to about 22, and then all new neuronal branching or arborization because of learning, and synaptogenesis, which is collinear when things are healthy. That is all going to be reflected in neurotransmitter and action potential patency. Now, CP1 and CP2 are found in microglia. We already know they're found in amygdala and prefrontal cortex. We went through that. They're found in microglia on the surface. And they have been implicated as playing a role for these processes, the same ones we just talked about there, neurotransmission, for example, that is by altering the neurotransmission of adjacent neurons, by altering their chemistry, by signaling, using endogenous cannabinoids like 2-arachidonyl-glycerol and, of course, anandamide, and accompanying synthetic and degradative enzymatic machinery like the fatty acid amide hydrolase. So this whole thing that regulates endogenous cannabinoid metabolism that you find in portions of the brain that we have studied is also found in the microglia. And microglia are neurons, so when these are altered in the microglia, what do you think happens? The microglia's function, again, as an immune cell and also as a sculpting cell to the neurons, and a cell that guides the neurons to their location once they're synthesized de novo as a neurogenesis. They're going to all play a role in that. And here we've got cannabinoid receptors there. Wow. That means that cannabis is going to have an effect on the neuroinflammatory response. Now, this gets a lot of people excited. It gets them excited because they say maybe by controlling neuroinflammation in the brain, by tinkering with the cannabinoid receptors in the microglia, we might be able to abort, alter, or completely stop neurodegenerative diseases, maybe even neuroproliferative diseases like glioblastoma. See, so there's a big hope there in the pharmaceutical industry, and indeed in the neuroscience industry. There might be something there by looking at cannabinoid receptors and maybe even exogenous synthetic cannabinoids. Maybe not the type that fully agonize and cause people to get inebriated, but maybe ones that would specifically target and alter the CB1, CB2 pathways in those microglia. Now, let me finish. Exogenous cannabinoids like THC, which are actually more potent, okay, so they bind more efficiently to the cannabinoid receptors than the endogenous do, but they're partial agonists, so that's a specific flavor in these exogenous ones, which means they don't fully agonize them. Doesn't mean they're not potent, though. It means they have a differential effect. But THC is a partial agonist, and they are not homeostatically regulated, though, as our natural ligands. So any alteration in arborization is caused by exogenous THC, for example, and the myriad of other cellular and molecular processes that we have just touched upon here that govern behavior and cognitive memory deficiency, which probably have neural correlates, could be immediately, that is short-term, and perhaps permanently corrupted. So this is something we need to study as a research community because we don't have enough data on it. We know a lot about ethanol. We know a lot about psychoactive drugs like the opiates, and we know a lot about the psychoactive drugs like cocaine and amphetamines, barbiturates. But we don't really, even though cannabis has been around for, centuries and centuries and centuries, we don't know much about THC and its effect on the system, and that's something we need to remedy. So thanks a lot for paying attention to this really long talk. I know it's probably pretty dark right now, but I let that happen naturally, so you see less of me. You hear more of me. The sun is setting here in the beautiful Northwest, which means we're getting close to the equinox because it's close to that time of day. Anyway, I'm going to end the show. Thanks for your attention to my talk, and I'm going to stop here by saying what I always do. I appreciate the time that you've listened to me. Please contact me if you have any questions or interest in furthering this discussion. I'd be glad to take it up with you. And I say unequivocally, bye for now.